For those who don't know me, I am Camilla. I am part of the On-Farm Carbon Advice team, and this is our webinar series. So thank you for joining us. Today, we have Ed Clayton, who will be walking us through the Low Methane Sheep project. And we have Geoff Casburn, who will be talking to us about supplement delivery systems in extensive livestock systems. So before we begin, I'd like to acknowledge the traditional owners of the lands and waters in which we gather today. I'm on Wiradjuri country in Orange, and I recognise their continuing connection to land, water, and the community. We pay our respects to their elders past, present, and emerging, and extend that respect to all Indigenous peoples here today. So this session will be recorded and will be available on our website where you registered for this. There is an opportunity for questions at the end of each presentation, but please feel free to put them in the chat and I will call on you at the end. There will also be a short survey at the end of the webinar series just to hear your thoughts about what we're presenting here today. So let me introduce you to Ed and Geoff. So Ed is a livestock research officer with the New South Wales Department of Primary Industries and Regional Development. Ed has worked in several areas related to forage production and omega-3 fatty acids, including forage quality, the effects of nutrition on reproduction, the impacts of diet and health attributes of meat, and methane production in sheep utilising mixed annual forage species for improved animal production and health. So Geoff is also based in Wagga and is a livestock development officer with the New South Wales Department of Primary Industries and Regional Development, and he's been with us for about 25 years. He's got a great knowledge of livestock and cropping systems and the interactions between these enterprises. He's very keen in livestock, he's got a keen interest in livestock production from pastures combined with sound drought feeding systems and lamb feed lotting. So without further ado, Ed, I might get you to share your screen.
No worries. Let me know when that comes up, Camilla.
That's perfect. You've done really well. We've lost your video, but that's fine. We can hear you, no problems at all.
Yeah, that's okay. Yeah.
Okay.
Might be easier, just bandwidth.
Sure, absolutely.
Okay, thanks. Yeah, we'll kick off.
Yeah.
First of all, I'd like to thank all the people that are involved with this project. It's quite a large project with MLA through the MLA donor company, UNE, and the Animal Genetics and Breeding Unit, which is a collaboration between DPI and UNE. We also have done a lot of work with producers on farm. We've worked with a lot of stud producers and commercial producers across New South Wales, Victoria, and South Australia, collecting data for this project with low methane sheep. So why is methane important from livestock? In this forum, it's probably not going to be anything particularly surprising, but livestock emissions account for approximately 70% of greenhouse gas emissions from the ag sector and 10% of Australia's total emissions. And sheep is about a third, quarter to a third of these emissions. So what we're aiming to do is to measure methane in 10,000 sheep across a range of genotypes and sire groups and examine genetic links for methane production. And combined with genotype and other phenotype data on those sheep, such as birth weights, live weight gains, reproduction traits, we're going to try and come up with, well, I'm not, the geneticists are, going to try and come up with a Australian sheep breeding value for methane. So a genetic progress in anything is slow, but it's a cumulative process. So they're going to try and reduce methane by 1% per year, but that's a 1% reduction every year, not just a one-off. And from an animal nutrition perspective, we like the thought of trying to reduce methane because if you have lower methane output, sheep are actually more efficient, because that methane just represents a direct loss of energy from their energy intake, and it can be 10 to 15% of dietary energy is lost as methane. So if we can reduce that, hopefully we make sheep more productive. We do this by essentially just putting sheep in a perspex box and measuring how much gas was in there before they were in there, and then how much was in there after a set period of time. This is a photo of one of our latest collections down in Portland in southern Victoria. It was a really nice setup, actually. We've got 12 chambers with a fiberglass water-filled moat to make it sealed, airtight, and we stick sheep in there for 40 or 50 minutes. Larger sheep actually measured for a shorter period of time because a larger methane output, a larger surface area in that box. And so these are just a few pictures of sheep in the chambers. I've been doing this for a few years now, and one of the things I was really interested to find when we first started was how well sheep handle being in the perspex boxes. Very little fuss going in. Once they're in, they just seem to stand pretty quietly. They can see their mates either side. They can see out. They're not particularly stressed about being in there, which is really nice, because we really need them just to breathe fairly normally. If they start panting or breathing too heavily, that can interfere with our ability to measure them. We use an analyzer called an Eagle 2 to measure the methane. It's a reasonably unique instrument in that it can measure a range of gases at the same time. It's actually used in the mining industry a lot for measuring sulfur dioxide and CO2 and a range of other things in mines. We measure methane as well as oxygen consumption and carbon dioxide emissions or output. And the reason we do this will become more important in a minute when we talk about what our measurements mean. And we work out a relative measure of gas as mils per minute. So we put them in there for 40 minutes, measure the gas before and after, take into account the volume of the sheep that was in there, and we can work out mils per minute. So it's not a total gas produced per day. So we can't use these figures that we've come up with for greenhouse gas accounting because it's not a yield of methane per day, but it's a relative measure, and we can rank sheep based on this relative measure. And when you look at that across a range of genotypes and sire groups, that's how you can start to select for sheep that produce less methane relative to others. There are a few different factors that affect methane that we'll talk about briefly. Time off feed or time after eating, the dry matter intake per day, and the weight and volume of the sheep. Temperature and pressure during the time of analysis, which we can measure and account for reasonably easily. This is some data from some information nucleus flock lambs at Kirby at UNE up in Armidale. We can see the time off feed and the amount of methane produced in mils per minute. If they've just had a big feed, there's more methane. As the time goes on, lower methane. So what we try and do is we try and standardise our measurement. We have them have ready access to feed up until an hour before measurement, and we take them off feed for an hour, and then we do our measurements to try and standardise that effect of intake. The timing of measurements is really important. So this will end up being a lot of info on this page, but we'll just step through it. The amount of time between putting the sheep in the box and measuring is very important. There are a lot of measurements that we have to take to account for background readings, standards and calibrations and things like that. So we've got our 12 chambers. We are measuring temperature and humidity in order to be able, and pressure, to be able to account for standardization of gases. The date and the run is very important, whether it's early in the day or late in the day, because temperature changes can affect gas readings. We take our first sheep off feed at 7 a.m. and then at exactly 8, it goes into the chamber, and then every minute and a half, another sheep goes into the next chamber. And that's because it takes about that long to do a reading about 20 minutes later. So we do our shed background reading then, and then 20 minutes after the first sheep went in, we do our first reading to check the accumulation of our gases, and that's done every minute and a half. And so our number 12 sheep goes in at 8:16:30, first readings at 8:20. That first lot of readings finishes at 8:36:30. The next reading is 8:40 through to 8:56:30. Then we do background readings again for the shed, and then we measure a range of standards to do our calibration. And so it's not a particularly onerous task doing the measurements, practically speaking, but we'll start at 7 or 6:30 or 7 in the morning, finish around 5 or 5:30 in the afternoon, and the longest break that you get during the day is about 10 minutes, or 8 to 10 minutes. So it's pretty constant. If you've got a couple of people doing it, there's not a whole lot of time for not actually doing anything. The next factor that's really important for methane output is intake. There's an equation that was developed by CSIRO with a compilation of data from everywhere. Methane output is 20.7 times dry matter intake, and that's what's currently used in our greenhouse gas accounting for ruminants in Australia. That came from Ed Clayton up in Townsville. And if we look at their data, there's an R squared of about 0.92, so 92% of the variation in methane was accounted for by intake. It's a pretty rubbery graph, this one, I reckon, because you've got smaller pasture-fed northern cattle on the lower end, and dairy cattle with really high intakes at the top end. It's pretty broad measurement. But we've got our own data from calorimetry experiments, which is a proper way that you measure metabolizable energy in methane for efficiency and livestock production point of view. And we've actually come up with a pretty similar relationship. 24 instead of 27, but this includes sheep and cattle. Same type of R squared. Our sheep right down the bottom, a little bit rubbery within sheep, a little bit rubbery within cattle, but it shows you that the relationship's pretty right. So what we're doing with our genetics is we're going to try and account for that intake, and then we're going to be working with the other, 8 to 10% of the variation. And if we can change that every year, that'll be a cumulative effect that we're talking about. So we try and account for methane with level of feeding. As we said, methane output's highly dependent on dry matter intake. We can't measure intake directly on the sheep that we are looking at on farm, but we can measure heat production. We can have an estimate of heat production, which is what I was talking about with calorimetry studies, where you're measuring sheep for 24 hours and you have a really closely controlled gas exchange, and heat production from this equation is related to oxygen consumption, carbon dioxide output, methane output, and while we can't measure urinary nitrogen, it's only a small impact, but we have all the other three inputs. So we calculate what the maintenance energy requirement of an animal should be based on its body weight. We use that heat production factor that we worked out from the gas consumption, and we look at a level of feeding factor, which is essentially multiples of maintenance energy requirement. And while it might be a little bit rubbery in places, we have live weight there and a heat production factor. It's not a bad relationship. You can actually account for a reasonable amount of variability in energy output using that. And if we look at our level of feeding factor, so multiples of maintenance, where maintenance is 1, you have a few issues with the data set just maybe topping out on methane. The R squared's only 0.58, but in terms of genetics and the variability that you can account for on farm, it's probably not a bad relationship sometimes. Doesn't always work. Here's a couple of other data sets from 2 different properties where it was obvious we weren't really capturing that level of feeding very well. A lot of clustering of data across different runs and different days, I suspect, in this one. But the geneticists assure me it's okay. All you got to do is collect more data and it'll all come out in the wash. So what do we do with analysis of data? Obviously, there's a lot of variation in methane within and between flocks, which is really good. You want variation because that allows you to be able to select for sheep that are higher or lower. This is an example of some of the output of methane across different properties in terms of the amount per day, what the average amounts are. Mostly it's kind of normally distributed across a flock, not always, sometimes a bit positively skewed, but it shows that there is a lot of variation. We standardise the methane output mils per minute with variance factors caused by feed and environmental differences, and look for fixed effects such as the flock, so the property, the run during the day. We do 7 runs during the day, and so, as I said, time of day will make it an effect. Birth type, sex, age, and that feeding level that I was talking about before. So there are preliminary results in terms of research breeding values and research heritabilities that the ag people have been able to come up with so far. In the current, the most current analysis, most up-to-date analysis, around about 5,500 records, predominantly from merinos, because that's what we've done a lot of our work in early on. I don't pretend to understand any of these genetic sort of values that they have there with phenotypic and residuals, but I do understand a little bit about what a heritability is, and it's, at the moment, they've come up with 0.19, which is pretty much smack bang in the middle of what you'd expect from any other heritability that they ever measure. Birth weight, growth rates, reproduction potential, all that sort of stuff. So it means that it's promising from being able to measure it. It's promising from being able to use that measure to make gains across different genotypes. This is kind of what it looks like when you put them all together. Sheep in this area are producing more methane than what you would expect from that flock average. Sheep in this area are producing less methane than what you would expect. And so they're in the process of coming up with project breeding values across our whole project with 10,000 measures. And as I said, if you have a negative project breeding value, you have less methane than what you'd expect. So all these measurements have been put into a database. We've got about 14,500 entries, some from our current project, some from past projects. Still quite a bit of data still to be entered. And what we are going to do with the current project is look at methane output with known feed intake. That'll allow us to calibrate some of our data that we've got with our level of feeding effect to try and take out a bit more variation between sheep and between flocks, with the aim to develop an ASBV either for methane output or methane intensity. They haven't quite decided what the best way to go is yet, surprisingly, whether it's grams per day of methane or grams per kilo of live weight gain or grams per kilo of lambs weaned, for example. We can reduce methane to 0 in sheep across Australia. You just don't feed them. Yeah, 0 dry matter intake, 0 methane, but that doesn't really help if you want to actually try and produce meat or wool. So I think, from our perspective, intensity is really important, and that's what we need to get people to think about. And, we've got to expand the database across other genotypes because it's mainly merinos at the moment. We ought to also look at other factors related to methane output. We've been analysing rumen fluid from a lot of these sheep for volatile fatty acids and bacterial populations. And if we can look at the relationship between those and methane, one, it might be able to give us a better measure of methane, or it might be easier to measure methane than actually having to stick them in a box for 50 minutes. And you can use volatile fatty acids to predict methane. Volatile fatty acids are fermentation end products in the rumen from when sheep and cattle digest starch and protein. If we have more acetic acid, we have more methane because those bacteria associated with methane producing bacteria. More propionic acid is more energy efficient and less methane output. And there are equations that people have developed in the past just to look at how those VFAs relate to methane output. And we apply those to some of our observations. This would be Kirby lambs, again, up at UNE. Even though your R squared's only 0.69, 70% of the variation, it's not bad. It's particularly not bad with some of the other estimates of genetic parameters that you might look at. So there might be good scope to include this at least in trying to get a more accurate estimate of what's going on in the rumen. So in terms of our project, we've measured methane around 9,000 sheep. Some more to go and some more feed intake measurements that we need to do. Research breeding values developed with a heritability of about 0.19, and we'll expand the database and examine feed intake and try and get a better estimate. So that's just about it. We'd like to thank MLA through the MLA Data Company, Merino Lifetime Productivity Project that's helped out with some of the collections, and all of our industry collaborators. We've been really lucky to have really good industry involvement. People are really keen on this project and offering up their time and their sheep to do measurements and collect all the data. So it's been really good.
Awesome. Thanks so much, Ed. That's great.
That's it.
Great. Your camera's still not working.
Yeah,
are there any questions?
No,
there you are. Nice to see you. Are there any questions from the room for Ed? Oh, maybe later. Maybe not. Okay, Geoff, you ready to rock and roll?
Hello, Camilla.
Hi.
Yeah, I am. Can you hear me?
Great. Yes, I can. Shall we share your screen?
We shall.
Amazing. Perfect. I can see me.
I can. Oh, here we go.
Yeah, we'll just reduce my face down.
I'm just wondering how. Here we go. Is that better?
Yes, and we might just. Is that in presentation mode?
Well, it is right now.
Fabulous. Yes.
Thanks, Camilla. And thanks, Ed. I always enjoy your presentations, and it's a lot of work that you're doing there. Now, I just want to introduce everyone to the low methane gang. Genetically selected, grazing low methane pastures, and fed additives to reduce methane. And it's the feeding additives to reduce methane that I'm going to be talking about today. Now, I'm not too sure, but I've just got a video to play here, and I'm hoping the audio will work. If it doesn't, Camilla, you'll just have to let me know.
No, there's no audio.
Righto. So basically, all over the country, sheep are getting supplemented, and they're doing that to either correct mineral imbalances or protein imbalances from low quality pasture. But also, there's the potential to use supplements as carriers for methane reducing additives. And I'll finish that there basically. And so today I'm going to talk about methane emissions reduction in livestock, which is called the MERAL program. And in particular, using electronic ear tags to assess and refine delivery of methane reducing additives in sheep. Now, I'm just a member of a large team. As we can see here, there's a range of us, and I'm going to be presenting on their behalf today. It's we have a lot of contributors to this work, and the New South Wales government and the Australian government, obviously climate change is a very high priority, and so they're contributing a great deal of money into this type of research. We're also lucky that we've got AWI, Australian Wool Innovations, and also some other industry partners are contributing to this work, along with universities, state ag departments and producers from wool producing states across Australia. Now, just recapping, Ed mentioned these first two dot points that I'm here, that 12% greenhouse gas emissions, agriculture contributes, and 76% of this comes from livestock. What's really important with these statistics is 95% of livestock production comes from grazing in extensive grazing systems. And that's where the biggest challenge lies. A lot of the work that we see done is in feedlot situations where we can really control what we're feeding animals. But in the extensive grazing systems, there's so many variables, regions, pasture types, times of year, that's going to be the real challenge. So the aim of our work is really to improve emissions intensity through more efficient use of animal health and production supplements, and that is less methane per kilogram of product produced, and also to reduce emissions through methane reducing additives. Now, there's a range of additives that can reduce methane. However, some of these additives that have the biggest impact on methane reduction may also have impacts on production. And so a lot of these additives, there's some challenges and there's some opportunities. Now, some of the challenges are that some additives need to be taken throughout the day. Some need to be fed carefully to prevent animal health issues. And some actually, like I said before, may impact on performance. Some of the opportunities, some are soluble. So they're actually suited to water delivery, such as water troughs. And some actually may improve performance, like I said. But the big challenge for all additives is that some animals eat too much, some animals eat the right amount, and some don't eat enough. And in extensive grazing systems, that is the biggest issue. So our work is actually trying to look at how we can increase the number of animals that eat the right amount. So what did we do? Well, we started looking at some of our common supplement delivery systems as carriers. So on the left-hand side, we've got a high volume supplement delivery system, a grain or pellet lick feeder. And on the right, we've got low volume, a loose lick feeder, which can deliver salt, lime, cause mag, or other minerals. And it's commonly known in the game that this is head down, bum up research. And really what we're trying to do is understand animal behaviour. When do sheep visit the feeder? How long is each visit? How many visits a day? How much do they eat per visit? And what is their daily intake? And really, it's really understanding what causes this variation or this behaviour that we're looking at. And so most of you would know who run sheep that it's now compulsory to have electronic ear tags. Now, electronic ear tags or radio frequency identification provides us with a great tool to help us start monitoring individual animals. However, the trouble is the technology has been developed pretty much to assess individual animals. So animals are either held in a crate or run down a single file in a race and ran individually. And so that enables the user to enter data individually on that animal. But what we need to do is we need to use this technology to assess multiple animals at once. And more importantly, we need to use it so we can actually measure how long animals are visiting a feeder. So there's some quite complicated maths behind being able to do this. And probably nearly all technology available on the market today doesn't allow this. But we've been fortunate enough to partner with two small businesses. One is the Feeder Reader from Victoria, and they have technology based on low frequency, and that's what we're using for NLIS for both sheep and cattle. But we also wanted to compare that with other options, such as UHF. And so there's another small business here in New South Wales, AgriScan, that have technology and readers able to use UHF technology. These two different types of technology have different strengths and weaknesses. One of the benefits of UHF technology is it can read multiple tags more easily at once, and it's actually used in the wool pipeline where in wool stores where you got wool everywhere in bales, you can drive down and read, basically do a stock take instantaneously. So what did we do? In terms of low frequency, we can see here we've got a common lick feeder on the left, and there's a big long panel across that feeder. Now that panel has a number of aerials inside it, which enables multiple animals to be read at once. The center photograph is of a water trough, so we can read animals when they come to the water. And the one on the right is a loose lick feeder that we commonly use as well. You'll notice they all have a solar panel because it's out in the paddock, and they have a control box which records that data and then enables that data to be either linked via LoRaWAN technology with a master control box or downloadable from USB. The second system we're assessing is ultra high frequency, and it operates slightly different. We can see here there's two little aerials along the top of this feeder on the left-hand side photograph. And if you can imagine what a spray formation looks like coming out of a spray driven behind a tractor, it's a cone shape. And so really those little aerials have a cone coming down, and as animals break that cone, they read the ear tag. Again, there's a big control box, and again, it's solar powered. What is quite unique with both systems is the technology needs to be able to say, okay, an animal is read by its ear tag, and it continues reading that ear tag. But we need to program when it cuts off a feed session, because if an animal moves its head out of the array for, let's say, 5 seconds, is it still counted as part of the feed session, or will it start a new feed session from when the last reading was? So there's a fair amount of work goes into understanding and I suppose optimizing aerial setup and read ranges. So what did we do? At Wagga here, we set up 4 pens with 6 lambs in it, and really this work was just to look at getting the two technologies working. We then progressed into small paddock trials with 45 lambs per paddock with 1 feeder for both low frequency and ultra high frequency. At Cowra, we did the similar work in the paddock with loose licks, but we also had some trail cameras, and we had the trail cameras set up in the feeder. So you can see number 4 animal here, we'd have the data that's collected by the reader, but we also have video footage of that animal accessing the reader. We had some cameras on fixed locations so that we could look at the animal's rumps and see which animals were visiting the feeder, and then again match that with the data that we collect. We also used Agilent ruminant, which is a methane reducing additive, either in the pellet form with the high volume lick feeders, but also sprayed onto salt at Cowra in the loose lick feeder systems. And the team at Cowra also did a lot of tag testing setup. So this is for the ultra high frequency that can read multiple tags at once. And so this box with tags was put under in a certain measured way under the aerials to assess how the tags were being read. And that was also done for the loose lick feeders. And what that enabled is a 3D diagram of where the read range was. So in this example that we can see here, this is for a signal strength of 240 decibels. And then we looked at adjusting the signal strength to 220 and how that changed the array. So really, this work is developing our understanding of using RFID to actually monitor animals in extensive grazing systems. With low frequency, it was a different setup because animals, there was a big long aerial and there was multiple aerials that could only read one animal at a time specifically, but there was a lot of aerials, so that enabled the system to read multiple animals at once. And this spits out an array as well. I don't expect people to understand what's this graph is saying, other than green is tag reads, yellow is partial reads, and reds are no reads. And so that measure was placed at different depths across the feeder and certain distances from the feeder, so we could work out what the read range was. So some of the results. What we've got here is we've got a graph that depicts UHF when we had 2 paddocks of UHF, and the black line on the graph is paddock 1 and the grey line is paddock 2, and these are dates down the bottom. So when we put the lambs in, we could see there was very few animals spending time at the feeder, which is the left-hand side of the graph. But as the animals became used to using the feeder, the time they spent there gradually increased to the point where it started to level out. Now, these 2 lines, while they're slightly different, they follow the same trend. And with statistical analysis, basically, UHF was reading in paddock 1 and paddock 2 on a similar basis. Along the top of the graph is a red line, and that looks at the gradient down the right-hand side, which is attendance. And pretty much on every day, close to 100% of animals visited the feeder. Now, as you can imagine, we had 4 paddocks of 45 animals accessing the feeder multiple times a day. So there was huge amounts of data. So members of our team, some of those members are really good at assessing data and also displaying it. And this is a great example of how you can look at some of the data and display it. Firstly, down the bottom of the graph, they're individual animal numbers, and the same animal in the bottom part of the graph is the same animal up the very top. So they're in line. And what we can also see is some of these bars are grey, dark grey, and some of them are white. The dark grey come from paddock 1 UHF, and the white come from paddock 2 UHF. And it's interesting just how the variation occurs across animals within each of those paddocks. What we can see, so firstly up the top graph, that's the average daily weight gain for all those animals below. And we can see, well, how does that average daily weight gain correspond with, let's say, the graph down the bottom, which is average time spent per day at the feeder? And there is some relationships there. And if we were to look at the right-hand side of the graph, a lot of the dark grey lines here were from paddock 1, and they corresponded with very little time spent at the feeder. And so they actually lost some weight during the trial. Where animals that spent a lot of time at the feeder often achieved some quite good weight gains, and there was a relationship there. If we look at the water trough, and we also look at the loose feeder work done at Cowra, in the top graph, it's really a time series. So we start at, let's say, 1 o'clock in the morning, and we travel all the way through to 12 o'clock at night. And we can see that animals access the water by and far the most period was at 12 o'clock during the day and through the afternoon. But we can see there's a relationship there with the bottom graph, which follows a similar time scale, but it's for the actual accessing the loose lick feeder. So there's a relationship between having a drink and taking a nibble of a supplement. So that's an area that we need to explore more in terms of understanding the behaviour and what things we might be able to do as land managers or in managing our animals to optimise supplement use. So what did we learn? Well, basically in our first work, that both low frequency and ultra high frequency technology works. Sure, there's some advantages and disadvantages between the two systems. And obviously within LIS, we are basically, most producers or all producers will have low frequency technology. So that's likely to be our main focus going forward. With both systems, there's also needs to be some further development required to manage large amounts of data and to be able to make sense of that data. Along with assessing RFID, we did do a little bit of work looking at supplement delivery. So again, like I said, we use some low volume loose lick feeders, and they can be good. They control daily intake by the concentration of salt, bitterness, or using actually various attractants to attract animals. And with high volume lick feeders, they control daily intake mechanically, using various lick mechanisms that regulate flow, placement, and feed gap. So I want you to have a bit of a look at this video, and at the end of my presentation, someone might tell me what we're looking at. Okay, so here's some photographs of how we actually adjusted the lick mechanisms on some grain and pellet lick feeders. Really, it's about adjusting the placement of where the animal sticks their tongue. And we can see some saliva here in that middle photo. And we can see in this feeder how we can adjust this bottom feed delivery system. The right-hand photograph is about a 5mm gap. And that at that gap, we across, like as an average, reduced intake approximately 750 grams a head per day intake. But we didn't do a great deal of work in this space, and so it's really just some very preliminary analysis. But when you restrict intake from animals and lambs, they get frustrated, like we would. And we can see here's a photograph on the left when there'd been some rain, and animals were obviously using their feet to try and trigger the grain to flow a little bit better. And so there are some problems. We can also see when setting a gap that it's often driven by the size of the pellet that's being fed. So we've got some lamb pellets here, and we've actually got some cattle pellets in the feeder. There's also potentially fines that can accumulate when using pellets, and that can restrict flow. And if we're using grain and we're having some sort of granulated additive like salt or lime mixed in, there can be some problems with those additives settling out. In short, look, it's early days and there's really much to learn, like electronic identification and having it set up in extensive systems is new. And we believe that we're going to learn a great deal from looking at how animals actually are accessing feeders. Some of the work that we've looked at, there is large variation in how often, when, and for how long animals visit feeders. Potentially, it mightn't be as easy as we think to restrict intake from animals, but it's about potentially the tongue becoming tired and walking off and regaining saliva again. We feel that, lambs that are trained to feeders will come onto the feeders quicker than lambs that are untrained. Mechanical restrictions may cause some unforeseen problems, as we saw with powdered and granulated additives potentially causing flow restrictions. Now, I only just touched on some of the results. Look, we produced 3 reports as part of Merrill 2. We've got a feasibility report, a field studies report, and a validation report. There are actually quite a lot of detail in those reports, and Camilla, we'll work with Camilla to make those available on the website. But And this work has really sort of led into current work as part of Merrill Stage 3 that we're currently embarking on. And there's a few different parts to that work. Firstly, we're just fine-tuning the technology to ensure that we can read a greater number of animals in the feeder at one time. The lambs that we used initially were 40 and 50 kilos. And so the maximum number we could fit in the feeder at a time was 9. But we want to make sure that the technology can read double that amount. Let's say we had 20 kilo lambs accessing a feeder to ensure that the technology can read greater numbers. We actually want to look at what impacts mob size might have on the variability of intake. So while we did 45 animals in our paddock trial, we want to look at what are the impacts of 40, 80, 120 and 160. And we want to do that with grain lick feeders. We want to do it with loose lick feeders. And we also want to look at lick blocks. And so that's going to be done at Wagga, Cowra and at Orange. We also want to look at the rate of intake. How much does an animal actually eat in a certain time frame? Because we are wondering whether, if we know how long an animal's eating, whether that can be how long they eat or how long they spend at a feeder, whether that can be a proxy for how much they actually consume in a day, which is really important when developing concentration levels in additives, but also in terms of in any sort of scheme moving forward in terms of reducing greenhouse gases. We're going to assess visitation patterns and how they may change when we use different additives. And we also want to do some more work looking at the relationship between water and supplement intake. And like I said, can time at the feeder be a proxy for potential intake? And I'm just about finished, other than I just want to mention a few collaborators in terms of research collaborators, specifically from UNE, University of Western Australia, and PIRSA and SARDI from South Australia. So thank you, Camilla, and I'll stop sharing.
Thanks, Geoff. That's so great. Ellie, do you want to come off mute and ask your question?
I can't see anybody.
So I'm happy to.
Amazing.
Thank you both so much. It was incredibly insightful. I had a question regarding like quantifying livestock emissions when it comes to climate disclosures and reporting. At the moment, I think regardless of any feed additive being used or particular genetic being bred, we're using a standard emission factor for methane emissions. Do you see this changing in the future? Is that sort of the plan, is that we could use a different emission factor depending on, the feed additives used?
I hope so. I hope that we do across a range of production systems and technologies. Geoff touched on low methane pastures. We have different pasture species that can reduce methane. Methane is not a set output. The level of feeding, not just dry matter intake, but the level of feeding compared with what you think maintenance is really important. So the efficiency of the rumen is important. I would like to see a lot more work going into better quantification of methane output, because that's what we really want people to do, is to be more efficient and get some better numbers on what your actual methane output is, just not a rubbery figure that's made up from dry matter intake, which might work sometimes, but it's not going to work in a lot of situations.
Yeah. Fantastic. No, thanks for that, Ed. Totally aligned.
Great. Thank you for your question, Ellie. Ralph, would you like to ask your question? Come off mute.
Yeah, sure. So Ed, great presentation, and thanks, Geoff, as well. The genetic correlations between methane production and productive traits. So fleece weight, growth rates. Is there any further information yet? I notice you've got the heritability, but those whether the correlations are antagonistic or together can make a difference on how much change we can make.
Yeah, we don't have all the answers yet, and that's the trick, because as I said, if you just select an animal for lower methane output, and we've got some preliminary data for this. You select an animal for lower methane output, they probably have a lower dry matter intake and lower productivity with either weight gain or lambs weaned. And so that's what the next stage of the work is going to be, pulling out all that info and looking at where the efficiencies are. There's a big assumption that if you don't produce methane, it goes into production, whether it be live weight gain or number of lambs. We're not there yet. We don't have all the info to say that's definitely the case, and how much of that energy is going to be partitioned back into meat or milk. But that's definitely where the focus is.
Great. Thank you, Ralph. Does that answer your question?
Yeah, no, great, thanks. Can I ask another one?
Of course you can,
yep.
yeah.
Just it'd be interesting just to feel for how stackable some of the technologies are. Like, will genetic improvement reduce the variation or the benefits, or increase the benefits of things like feed additives or pastures with plant secondary compounds?
That's a good question, because you select for one thing and you don't know what it's going to do on another thing. We're hoping that we're going to get additive effects, as Geoff pointed out in his first slide. If we have a low methane pasture and a sheep that's genetically predisposed to not producing as much methane plus an additive, that's where we're hoping to get the biggest gains. We're certainly not thinking that any one technology or any pathway is going to lead to all the answers. It's a multi-pronged attack that's going to, a bit like integrated pest management. A multi-pronged attack is going to give you the best answer. But you're right. You select for an animal that's genetically lower in methane output. What does that do to its response to some other factor? Unfortunately, we don't know yet.
Great. Thank you, Ed. Thank you, Ralph. Final questions from anyone else in the room?
Stephen Hobbs in the chat.
Yeah, great. Stephen, do you want to... Stephen, do you want to ask your question? Or Ed, are you just happy to respond?
I'll just see if I can...
So the question is, do you think that sheep that maintain their weight more consistently over a given frame would be more feed efficient, therefore lower methane output?
It's quite possible that they are. It is quite possible that they are, and that's the sort of stuff that we really want to measure. Yep.
Fabulous.
Thanks for asking the question. Sorry, I was busy trying to find the button to speak, and I'm on.
That's okay. No problems at all. Do you have any other questions?
I've got a question. Does anyone know what that video footage was of? I don't know. I have a thought. Yes, Stephen took himself off mute. What was that little thing with the pellets? No? Geoff, you put us out of our misery. Well, look, it just happened to be that that was footage of inside a grain lick feeder. And so that was the animal's tongue outside, poking it in that little gap that we were showing. And really the importance of that footage, it seems it's really quite interesting, but it's just how the tongue picks up the grain to feed. And so if you restrict that, and there's a range of designs out there which control that, and it's getting a better understanding of how we can control those animals that eat too much. If we can reduce that, because what we've got is we can measure disappearance of a supplement that we provide, but we really don't know how effective it is across the mob. And we want to do that. We want to reduce the variation as much as we can, so that there's some other measures which will give us a bit more accuracy. So it was just some interesting footage of the tongue.
Absolutely. Henry thought it might have been one of your kids inside the lick feeder, but there you go.
If it was an ice cream, probably.
Yeah, exactly. Okay, thank you very much. I am going to wrap it up there with 3 minutes to go. I just want to say a big thank you to Ed and Geoff for carving out some time and presenting their research to us today. I found it really, really informative. Has been recorded today, so it will be available on our on-farm carbon advice webinar series page. I've also put a small survey. It'll take you less than a minute to fill out. It just gives us some really good feedback about whether or not this was informative or at the right level of information. Our next series will be the Low Methane Beef project, and that'll be on the 30th of July. So feel free to register for that event. And as always, if you want to give us direct feedback, please email us at carbon@dpird.nsw.gov.au. Fabulous. Thank you again.
Thanks, Camilla. Thanks, everyone.
Thanks, Camilla.